Hello, everyone. I'm Dr. Carmen Pugliafito, and I'm here today with Professor David Zacks of the Kellogg Eye Center at the University of Michigan. David, welcome back to Retina Synthesis.
It's great to be here. Thank you so much for having me. Appreciate the invitation.
So, you pursuing a novel approach to the treatment of geographic atrophy. Can you tell us about the biology of your approach?
Sure, absolutely. My research actually started in trying to understand why do photoreceptors die during retinal detachment. This was something I started during fellowship and continued as I became a junior faculty here at Michigan. And what became clear was that the FAS receptor, FAS receptor, plays a key central role in regulating cell death during retinal detachment. But what we didn't appreciate in the very early days, but soon became clear to us, was that what we really had hit upon was a core common pathophysiology, and that affects and regulates cell death in the retina across a variety of disease states. We began testing different, we had originally had a retinal detachment model, but then we started using animal models of other retinal diseases. And one that really was very intriguing to us was macular degeneration, AMD, and particularly the atrophic form. And what we found was that FAST was very important in regulating cell death in that context as well. And um, just as an aside, it also was found to be very important. And this was work that we had done in collaboration with investigators at Scapins, Meredith Gregory Cassander and her team, showing that it was very important also in regulating ganglion cell death in open-angle glaucoma. So what we were really finding was that this was a core common pathophysiology. And it made sense. Why would cells evolve multiple different ways of regulating cell death in the retina? This kind of made a lot of intuitive sense. And then when we thought about it even more, one of the compelling features of targeting FAST was that it was very upstream. When you activate FAST, two things happen. One is you initiate cell death pathways, the apoptotic cascade, and that's well understood and has been recognized for many years. But a second thing, and I think this is actually equally as important as the activation of the cell death pathways, but when the receptor gets activated, you're actually also turning on the inflammatory cascade. And it's inflammation, or this para-inflammation, as some people call it, which is this very focal inflammation, that this is what's important in activating the innate immune system within the retina. You get microglial activation, proliferation, and then migration to the site of injury. And it's this inflammation that's important in propagating and perpetuating the disease state. And what we found in our animal models was that when you block the FAST receptor, and we did it in a multitude of different ways, genetic models, small peptides, siRNA, neutralizing antibodies, all these different ways of blocking the FAST receptor. But what we found were two things. One, as expected, it blocked the activation of the death pathways. But secondly, it also quieted down the inflammatory response. And it was really that one-two punch, we think, that kind of helped separate the fast receptor mechanism of action from kind of targeting just death pathways or just the inflammatory cascades. So that's kind of in a nutshell the history of our work in FAST in the retina and why we are targeting FAST in retinal disease.
So, you have a peptide.
We're using a small peptide. This is a 12 amino acid peptide. It's delivered intravitreally. The history of this is interesting. It was actually the parent compound of the one we're using now was originally developed for liver, for regulating liver damage. Hepatocytes die in acute hepatic injury through fast-mediated apoptosis. One of the problems was that the team that was developing that, um, they could not deliver it to in measurable quantities to the liver. As soon as this peptide was delivered into the bloodstream, it was broken down by peptidases. We tried this and they published their results. We saw this peptide and we injected it into the eye. And we saw that, the eye being kind of a privileged space, it worked there. And it actually had pretty good kinetics and pharmacokinetics. So, it was a viable alternative. And one thing that we've subsequently shown over the years, we developed then this next generation peptide, the one that we're using now in the clinical trials. But a key aspect of it is that as soon as it leaves the eye, it gets broken down very quickly in the bloodstream. And so, there's always concern about blocking something like FAS, which, is involved in many, many different pathways, and there's concern about tumor formation if it escapes the eye. It's a key safety feature of our drug. It's broken down immediately in the bloodstream.
How long does it stay in the eye? What's the half-life?
The half-life is quite long. So we have about 100-day in our current formulation, about 100-day half-life. And that's what is allowing us in our clinical trial, which we'll get into shortly, 3 to 6 months dosing. So we have good durability in the eye.
So that's attractive. So tell us about the clinical results.
Yeah. So one of the key things, so we've done clinical trials with our current peptide in three indications. We did phase 1 and 2 in macular off-retinal detachments. We did a phase 1 in glaucoma, open-angle glaucoma, and a phase 1 in geographic atrophy. In all of these cases, we're seeing very strong signals for efficacy. And now we've just recently launched at the end of 2025 a very large phase 2 trial, the Galaxy trial for the use of this peptide in geographic atrophy. And what gave us the confidence to go into this large trial? So I'll kind of summarize the results of our phase 1 trial. In the phase 1 trial, we had two components. First, we had an open label dose escalation. These were 6 patients, 2 at 50, 2 at 100, and 2 at 200 micrograms. There were very strict escalation criteria based on safety measurements. And these patients received a single injection of ONL-1204 and were monitored for up to 6 months, for out to 6 months. And we didn't have a sham group in this, but we did have the fellow eye as a comparator. And we saw that, first of all, it was safe, it was well tolerated, there were no problems giving this injection. This was the first time we had given an injection of ONL-1204 without a vitrectomy. In the previous retinal detachment study, we had given the drug and then it was removed during the course of the vitrectomy for the retinal detachment repair. Here, it was not removed, and we saw that it was safe and well tolerated. Interestingly, when comparing the rate of the GA lesion growth in the study eye versus the fellow eye, we saw about a 42% reduction with a single injection measured out to 6 months. And that was very encouraging. That was really, really exciting. Then we proceeded into kind of what we call the natural history treatment phase of the study. This was the second part of the phase 1 trial. In this part of the trial, we enrolled patients and monitored them, and these were patients with primarily fovea involving, but some were non-subfoveal GA lesions. And the inclusion criteria were very similar to the Apellis trial in terms of lesion size and such. But basically, we monitored these patients for about 6 months, looking at their rate of lesion growth. Then they were injected with ONL-1204, low dose, high dose, or a sham. So they were randomized into one of those 3 groups. And then 3 months later, they received the second injection and were monitored for an additional 3 months. So 2 injections, 6 months. And what we saw was that ONL-1204 compared to sham slowed the rate of lesion growth by depending on in a dose-dependent manner with the higher dose, reducing lesion growth by about 50% with 2 injections measured out to 6 months. So that was really exciting. In the open label dose escalation, comparing the study eye to the fellow eye, about a 42% reduction. And with the higher dose in the natural history treatment phase, 2 injections slowing the lesion growth by about 50%. And again, it's 6 months, measured out to 6 months. So very exciting preliminary data, encouraging, and give us the confidence to go into this large phase 2 trial.
So how were you measuring lesion size?
Lesion growth, FAF, fundus autofluorescence,
Mhm.
very traditional way.
Were you surprised by the extent of inhibition?
Um, Surprised, ecstatic, thrilled. Surprised, I think, this we want to be very cautious. Um, this was a phase 1 trial. We're now trying to replicate these results in a larger 325 patients phase 2 trial. Um, but I'm very optimistic. I think mechanistically, it makes a lot of sense. We always like to say, does it pass the sniff test or, if you do a gut check. So remember, I told you that FAST does two things. It activates cell death pathways and it activates um, inflammation. We think that by targeting those two pathways, we are really able to have kind of an additive effect and not just, and that might allow us to have kind of an outsized effect based on our mechanism of action. One thing that I didn't mention in our results, but I think is important noting, we also saw the potential for visual improvement. In the natural history treatment phase, on average, those 6 patients in the study eye versus the fellow eye had about a 5-letter gain of vision. And in the second component, the natural history treatment phase component, where we slowed lesion growth by about 50%, over the year of the patients being in the study, the sham patients lost vision, lost letters. And in the ONL treatment group, after treatment, there was about, in the high dose, about a 5-letter gain compared to baseline. So there seems to be the potential for actually improving or maintaining vision by blocking the FAST receptor. And that also, I think, speaks to the inhibition of the inflammatory cascade. There's kind of a spectrum of cell uh, physiology. Some are healthy. Those are the cells far away from the lesion. Those in the very center of the lesion are probably dead. But there's a spectrum of cells as they're kind of approaching death. And by reducing the inflammation, blocking cell death and reducing inflammation, we allow the cells that are not dead to kind of rejuvenate, resuscitate those cells, and allow them to do what they're designed to do, which is see and read letters on an eye chart. So from a mechanistic perspective, we think this also makes sense and is consistent with our understanding of the biology.
So there are some advantages here over the complement inhibitors, correct?
Potentially, we think that there's the potential for, and again, this is what we're trying to prove out, but we think that there's potential for improved dosing. In a phase 2 trial, we're having 2 dosing arms in terms of duration, every 3 months and every 6 months. So that would be very attractive relative to the current complement inhibitors, and also the potential for visual improvement.
What about the inhibition regulation? Similar to the complement inhibitor drugs or better?
Inhibiting lesion growth.
Lesion growth.
Well, again, this has to be proven out, but if we really have this outsized effect, I think it would be potentially better. But, that's the goal.
Tell us a little bit about the Galaxy trial that's underway right now.
Yeah, absolutely. So the Galaxy trial is a very large phase 2 trial. We're hoping to recruit about 324 patients. This study is being conducted in the United States, Canada, and in the European and in Europe, United Kingdom, um, Switzerland, and a number of other countries across the EU. We are randomizing into a low dose of ONL-1204 and a high dose of ONL-1204 every 3 months, and the high dose of ONL-1204 every 6 months. So we've got 3 treatment arms. It's compared to a sham group. And in the United States, we have a comparator arm against Izervay. So, obviously in Europe, these are not the anti-complement and the drugs are not approved. So that's why we have the sham group. But in the United States, we have the reference arm against Izervay. The endpoint, the primary endpoint will be lesion growth of ONL treated versus sham at 12 months. We will carry the study out to 18 months, primarily for durability and safety.
Well, this is wonderful. When do you think the last patient enrolled will be?
Oh, it's never seen enough, as you know. We are actively recruiting. We're thrilled. We're up and running in the United States and Canada, and United Kingdom and Switzerland were just recently activated, and we're awaiting final approval in the European Union. We hope to get that in early March, and we'd love to have recruitment done by the end of 2026. So.
That'd be, well, this is an exciting new development uh, comparing to complement inhibition and some significant advantages which we've outlined. David, congratulations on your work. It's been a long road, but it looks like you've made some significant advances.
Well, thank you. No, it's a thrill to be working in this space. It's a thrill to try to get something from the lab into the patients, and you know, by studying the basic biology, hopefully we can make a difference. I appreciate the
Okay.
opportunity to speak about it.
Thank you very much.
Appreciate it. Thanks, Carmen. Take care.